Hello students, myself Popere KS, Assistant Professor Botany of Rare Shikshan Sanstha's RBNB College, Srirampur. Now in this lecture, we are going to study about the repair mechanism of DNA or the DNA repair system. So, what is mean by DNA repair or the different DNA systems which are present in living organism? So, the DNA molecule, it should be protected from the physical and the chemical damages or the UV rays or the harmful gases. So for their protection, there are different types of mechanism which is present in living organism. So this will allow the genetic material to be saved from the damage, and so it should have, as the genetic material should have high fidelity, so there are different repair system present in all living organism. There are different enzymatic reaction which are taking place in the organism which act as a repair system. Now this repair, when will this be occurring in a cell? So this repair system or the repair mechanism, this will be occurring in the replication stage. So this repair mechanism, it will be occurring it is occurring prior to replication or operating in the replication stage, and some also will occur after the replication stage of the cell. Now we will see the different types of replication, that is the repair system present in the living organism. So they are broadly classified into two, that is the system which operate in presence of light is light repair system, and one more is the dark repair system in which there is no involvement of light. So first is light repair system. And the second one is dark repair system. Now first we see about the light repair system. This light repair mechanism or the light repair system, it is occurring in presence of light. So it is also called as photo reactivation mechanism. This first one, we will now see the first one, that is the light repair system. So this light repair system, it is also called as photo reactivation mechanism. Now what will happen in this type of repair mechanism? In this, suppose there is a formation of thymidine dimer due to exposure of DNA to UV. Due to heavy dose of UV, there will be formation of this thymidine dimer. Now what will happen during this photoreactivation mechanism or the light repair? There will be involvement of one enzyme, that is photolyase. So what this photolyase will do? This photolyase, it will recognize the thymidine dimer. So firstly it will be in the inactivated state. After exposure to the visible light, that is so in presence of light, now this photolyse will get activated and it will recognize the thymidine dimer. So it will come and bind here. So this is the photolyse enzyme, and what now it will do, it will break the thymidine dimer or the covalent bond between the thymidine dimer. Now what will happen, this DNA will again regain its shape. So now it has again come back to its normal state. So photolyse enzyme in presence of the visible light, it will break the covalent bond between the thymine or the thymidine dimers, and the this repair so is again this repair is resolved, and DNA has come back to its state again, and that is to the normal state. So this is the light repair system or the photo activation mechanism. This enzyme is also called as photo reactivation enzyme. That is photo reactivation enzyme, it is photolyse. Now next we will see about the second, that is the dark repair system. In dark repair system, there are different types of repair system that will be occurring. So first is, we will see there is involvement of different enzyme in the dark repair system. First is base excision repair. So what is happening is base excision repair. There is removal of one nucleotide, that is the wrong nucleotide, which get incorporated into the DNA. So in this it will occur with the help of the enzyme, that is which will remove the wrong incorporated enzyme. Now here what will happen, it will occur in the G1 phase of the cell cycle. It is occurring in the G1 phase of the cell cycle, and there is removal of the one nucleotide, that is the the one which is wrongly incorporated. The one nucleotide will be removed. So first it is recognizes by, suppose so this is the wrong nucleotide which is incorporated, that is hypoxanthine. So what will happen, there will be one enzyme, that is glycosylase. It will recognize the mismatch site or the wrongly incorporated nucleotide. So now this glycosylase enzyme, which it will means bind to the wrongly or the mismatch site, and it will break the glycosidic bond between the nucleotide. After that, now there is a purinic site, that is AP site will be created, because as this nucleotide is removed by this glycosylase, now AP site is created, that is the apurinic or apyrimidic site is created. Now what will happen? Now after this DNA polymerase, it will come and it will synthesize the or it will fill the gap which is created by the glycosylase enzyme, and now ligase will seal the gap. With the help of this template, this DNA polymerase is going to synthesize the new nucleotide where the gap or fill the AP site, that is apurinic or apyrimidic site, and ligase will seal the gap. So this is the type of, that is base excision repair. Now it is a type of dark repair system. Now next is one more is there, that is the nucleotide excision repair. Now what will happen in this type of repair? In this, it is occur in the G1 phase of the cell cycle. There is removal of 2 to 5 nucleotide, means comparatively longer sequence of the nucleotide or a long length strand will be removed, that is 2 5 nucleotide will be removing. So now in this case what is happening, suppose this is a site here, but suppose this adenine, it is in its immunoform, that is there is some changes in the nucleotide, that suppose this A is in the immunoform. Now what we'll have to do, now there are certain enzymes which are going to do this repair mechanism. So firstly there will be a UVRA. Now what will this UVRA will do? UVRA will come and it will direct UVRB. Now what will happen, suppose this is UVRP and these are UVRA. Now this UVRA, they will direct the binding of UVRB to the wrongly incorporated or the mismatch. Now what will happen? Now due to this there will be bending of the DNA. Now there will be this is bending is seen, that is in 9 nucleotide. So here in this due to binding of UVRA and UVRB, there will be bending caused in the DNA. Now next what will happen to this UVRB? Now one more enzyme will bind, that is UVRC. Now this is UVRC, this will bind to UVRB. Now what will happen? This will cut at 5' side and this will cut at 3' side of the DNA. Now after this, now they are going to create a nick on the DNA. After that what will happen? One endonuclease will come, that is UVRD will come. It has helicase activity. Now what will happen? This after the cut, this UVRC and UVRB, they will be removed. Now there will be involvement of UVRD. Now this has helicase activity. It will unwind the DNA at this place. Now after that endonuclease will come and it will remove the wrongly incorporated nucleotide. After that, DNA polymerase third, it will fill the gap, and one more enzyme, that is ligase, it will seal the gap. So this is nucleotide excision repair. There are different enzymes which are involved in the repair mechanism, that is UVRA, UVRB, UVRC, UVRD, then endonuclease, DNA polymerase third, and ligase. Now next we will see the next mechanism, that is the mismatch repair system. This is also a type of dark repair system. What will happen in this? This will occur the wrongly incorporated nucleotide, that is which is cause of the tautomeric shift, so that it will be repaired by this mismatch repair. Now what will happen in this mechanism? There is here is wrongly incorporated nucleotide. Now here also some enzyme will play the role. So first there is involvement of mut-S. So this is mut-S, which will incorporate one more enzyme, that is this is mut-L, and it will further allow the binding of one more enzyme, that is mut-H. Now in this mismatch repair, what will happen? Along with the wrongly incorporated nucleotide, some other nucleotide will also remove along with this mismatch nucleotide. So what now? This mut-H, it has helicase activity. So what will it will do? It will remove break the bond, the hydrogen bond between this nucleotide, and now what will happen? It will cut from, that is 5' to 3' side. So what will happen? There will be the removal of the entire strand, that is along with the wrong nucleotide, other nucleotide will also be removed. Now then there is one endonuclease enzyme will be there. Endonuclease enzyme, which will remove the nucleotide strand. After that, DNA polymerase will come, that is DNA polymerase third. It will incorporate the new nucleotide, and one more enzyme, that is ligase. It will seal the gap. So firstly, here the wrong nucleotide, it is recognized by mut-S, which will allow the binding of mut-L, then mut-H will be bind. Now here there will this mut-H, it will cut at 5' to 3' end. Now endonuclease, it will remove the strand, that is the this strand of the DNA along with this wrong nucleotide, and DNA polymerase third will bind and synthesize the DNA, and ligase will seal the gap. Now after this, one more repair mechanism is that is recombination repair. This recombination repair, it mostly occur when there is extensive DNA damage which is occurring in the living organism. So during this, what will be happen? A long strand of the DNA or the Suppose this is the DNA. So during this recombination repair, there is extensive damage, and the damage may be present in both the strand. So in that case, what will be happening? The repair machinery is going to work, and it will scan the whole DNA or the entire DNA, and it will see the similar sequence which will be similar to the lost part of this DNA. And now what will happen, that a sequence will be copied and they will now transfer to this region. So this repair mechanism or the recombination repair mechanism, it occurs when there is highly extensive damage occur to the DNA, and this damage system is also called as retrieval system. It is also called as retrieval system. Now after that, one more damage is there, that is source. So this repair mechanism or this repair system, it is also called as save or serve. Why? Because when there is high extensive damage occurring in the DNA, this may be due to the formation of thymidine dimer in the DNA. So what will happen, because of this thymidine dimer, what will happen, there will be that is the DNA polymerase can't work, or the repair machinery which are available in the organism or in the cell, the different repair machineries, they can't work, because this thymidine dimer, it has blocked the DNA polymerase and it will not allow it to work. So what will happen, there will be involvement of certain different types of DNA polymerase, that is suppose DNA polymerase 5 or 6. Now what this DNA polymerase do, they will incorporate any nucleotide to the DNA strand. Why? So it is also called as error prone repair system. Because as there is incorporation of any nucleotide to the DNA strand. So saving or solve means this is the last step, so which can save the DNA, or else what will happen, due to the DNA damage, the cell may die. To save the cell, this mechanism is there in the cell, which is also called as error prone repair system, because there are chances of error which will be incorporated into the DNA. So this is the last type of, that is SOS repair. So what we have studied about the different repair mechanism in the cell, that is the light repair system and the dark repair system. In light repair system, there is involvement of one enzyme, that is photolyase, which will break the thymidine dimer in the presence of the visible light. It get activated and it will remove the thymidine dimer. And in next, that is in the dark repair system, there are 5 different types of repair system, that is base excision repair, nucleotide excision repair, then mismatch repair, then recombination repair, and the last one is SOS repair. Okay, thank you.